Assalamualaikum and good day, everyone. I'm Mr. Ahmad Fawzi, the lecturer resource person of PHC415 Pharmacy Practice. Today, I'm glad to introduce to you the dispensing lab session and compounding tests so that you can better understand the learning flows. Let's get started. I would like to disclaim that some part of this presentation belongs to Nur Sabiha Mat Hussein, our previous resource person, PHC415, and I am glad to use some of them. Our labs begin in week 8 onwards, while compounding tests will be scheduled on both week 10 and 13. All sessions will be held in dispensing lab 1 until 4. Kindly note that the different duration of each type of learning activities, and these two activities are individual work. Here is a picture of lab session. Everyone is wearing clean lab coat as instructed, so please check your attire. Here are the directory if you lost within the faculty area. FF2 block is for tutorial, FF3 building is for labs and compounding tests. Always check the venue for each. This is BB8. So in lab sitting, we use code. So what's your code? In order to determine your seat, first check the table of lab or tutorial grouping. From there, look for your group and number assigned on the name list. For example, B1 group and number assigned 12 means your seat code is B112. So here is B112, B1 from your group and 12 from your assigned number. From there, you can check date, time, and dispensing lab venue, as well as the lecturer in charge. By referring to the floor map of the dispensing lab 2, you can immediately know your seat by looking at the code B112. Remember, you are not allowed to change any of the arrangement. So what are you going to bring during the labs? You can see a list of references and books. Remember, some are not allowed to be brought in compounding tests. We also have a limited copy of BP and BPC 1973 for sharing during the session. Apart from that, you also have to bring all these items, and remember, print your lab manual, worksheet, and rubrics sufficiently so that you can send them at the end of the session. During compounding, make sure you minimize your movement, work in clean and tidy manner. Whenever spillage occur, always wipe it cleanly. Safety first is our mantra in this case. Whenever you arrive in dispensing lab, you will see that you have your own working space, apparatus, some weighing equipments, as well as hot plate for you to compound the medicine. Learn how to use them and use it safely. Remember, some of you might have to share because of limited number of availability. During lab session, you have to prepare your worksheet and of course your dispense medicine. You have to work individually, and at the end of the day, you have to send both items so that we can give you mark. Of course, whenever you compound medicine, you will see pharmaceutical calculation. Always do it carefully and try to relate with the practice. And remember, always double check every calculation. In order to minimize the error, whether in compounding or in calculation, you have to do it in an organized manner. Okay, so you have to write clearly, double check, write down everything, and do not take any shortcut. Do not rely 100% on your calculator because sometimes you can always check using your brain. So how do we prepare worksheet? First thing first, you have to ask all these questions. Okay, for example, for internal or external, it will determine the selection of label and container. In the case of intended to be used for patient or for stock storage, then labeling preparation might be different. In the case of official or non-official formulation, these will carry different label preparation. So you have to refer all these to your Introduction to Pharmacy Practice lecture notes for details. Whenever you find any underdose or overdose, always rectify or speak to your lecturer, who is usually a registered pharmacist. Get the endorsement from him or her and then work immediately. Always follow all the step-by-step procedure that presented on this slide so that you can get a correct compounding preparation steps. So how do we mark your worksheet? Remember, each carry 15 marks. Basically, we will look at the major mistake and minor mistake. The major mistakes will be a deduction of marks of 10, while minor mistake will be deduction of 1 mark for each. And this is actually negative marking. You start with full marks, then we deduct gradually until you left with 5 marks. Okay, let's have a look at the major mistake. We only deduct 10 marks only for once. Whenever you have more than 2 or 1 major mistakes, we will no longer deduct or penalize. Meaning to say that we will have a maximum deduction of 10 marks which carry 1 major mistake. While in minor mistakes, whenever you did not do any major mistake, we will deduct 1 mark each for every minor mistake. Also, it has maximum deduction of 10 marks, which is for 10 minor mistakes. So all in all, you will left with 5 marks in the end of the day. That's all. Thank you very much for your time, and I hope that you are prepared for your lab session as well as compounding test. And if you have any question, please do contact me through email or through our YouFuture platform. Thank you very much.